Good morning to everyone. Myself M.Suganya, Assistant Professor at Department of Biochemistry in KSR College of Arts and Science. Today we are going to discuss about haploid plants. Before enter into the topic, what is haploid plants? Cell or some microorganism is set of chromosomes like haploid. Haploid plant formation, there are two methods are involved. In first step, in haploid plant formation, two methods are involved. First one, androgenesis, and next one, cyanogenesis. First method, in vitro methods, that is androgenesis. It is androgenesis within the production of haploid plants from microspores. Anther culture for production of haploid plants, it's reported in 250 species. Then Salmonaceae, Cruciferae, Germinaceae, Ranunculaceae, most common species are involved in androgenesis. The next method, cyanogenesis, production of haploid plants from unfertilized egg cells. Egg cells are involved in cyanogenesis. So, two method steps are involved in haploid plant formations. Next, what are the applications are involved in haploid plant formations, like development of homogeneous line, next generation of exclusive male plants, then induction of mutations, then production of disease resistance plants, production of insect resistance plants, production of salt tolerance plants, cytogenetic research, induction of genetic variability. So, these are the applications, main applications are involved in haploid plant productions. Then, somoclonal variation. Somoclonal variation means genetic variation in particular plant that have been produced by PTC, plant tissue culture, and can be detected as genetic or phenotypic traits. So these are the definition of somoclonal variations. Next, applications of somoclonal variation crop improvement. So many applications are involved in somoclonal variations, like first selective novel variant. First we have to select that novel variants, then disease resistant, abiotic stress resistance, then salt tolerance, then aluminum tolerance, herbicides resistance, insects resistance, seed quality. So, these are the main applications of involved in somoclonal variations in that PTC, I mean plant tissue culture. Micropropagation has many in vitro culture we have to handle in nowadays, like tissue culture, mass propagation, in vitro culture, cloning method. So, these are the main propagation, micropropagations are involved in the PTC. The in vitro clonal propagation. In vitro clonal propagation, micropropagation is rapidly multiplying stock material to produce a large number of progenic plants. Using modern plant tissue culture methods are involved in that in vitro clonal propagations. Widely used, many interior photogenic plants are involved in that micropropagations, in vitro culture techniques. We have to handle that micropropagations. So, in that micropropagations methodology, major steps are involved in micropropagation. In first stage, in that methodology, selection of mother plant and its maintenance. That is the first stage. In that next stage, initiation and establishment of culture. So, this is the first stage. And the next, multiplication of shoots or rapid somatic embryo formations are involved in that methodology. That is second. In third, in vitro germination of somatic embryo in roots of shoots. It's involved in roots or else shoots. So, this is the third stage. And that fourth stage, mother plant, transfer of plantlets to sterilized soil for hardening under greenhouse environment. So, these are the methodology steps are involved in micropropagations. Next, applications. What are the applications are involved in micropropagation? Let's see about somoclonal variations, then germ plus conservation, mutation breeding, then inducing mutations, embryo culture, haploid and diploid plant productions, in vitro hybridization, protoplast fusions, disease-free plants, then molecular forming, genetic engineering, production of secondary metabolites like alkaloids, flavonoids, terpenoids. So, see, these are the secondary metabolites are involved in the micropropagation. So, these are the main applications of involved in that micropropagations. So, next we are going to discuss about plant tissue culture. So, what are the applications are involved in that PTC, like see, clonal propagations, then large scale multiplication, breaking dormancy, secondary metabolites I already told, like flavonoids, terpenoids, alkaloids. So, these are the secondary metabolites are involved in that PTC culture. Then wide hybridization, genetic variability, disease free plants, then haploid, polyploid, terploids. These are the applications are involved. International of exchange germplasm, somatic hybrids, synthetic seeds, genetic transformation. So, these are the applications are involved in that plant tissue culture. Now, CAM cycle, cranulation acid metabolism, it is otherwise named as C4 cycle, CAM pathway. So, these are the names of CAM cycle. Two steps are involved. Open stomata, closed stomata. Night time it will be closed, stomata will be closed, open type, open. CO2 it's enter into the mesophyll cell. Open stomata permits of CO2 and loss of H2O. Phosphoenol pyruvate it's converted into triphosphate, again it's converted into starch. That process are involved in chloroplast. In that oxaloacetate is converted into malate is converted into malic acid. That the process are involved in vacuole. At the time enzymes or malic acid dehydrogenase involved. NADH plus it's converted into NAD. So, these are the steps are involved in open stomata. CO2 uptake and fixation, leaf acidification is involved. This is the open stomata. Next day, closed stomata. Closed stomata prevents H2O loss and CO2 uptake. Enter into the Calvin cycle. At the time malate is converted into pyruvate is converted into starch. This is the process are involved in chloroplast. In that malic acid vacuole, inside the malic acid it came out into the chloroplast. At the time decarboxylation of stored malate and fixation of internal CO2 deacidifications are mainly involved in CAM cycle. Next, C4 plants preventing photorespiration. In that C4 plants it's otherwise named Hatch-Slack pathway, that is the Calvin cycle. PEP carboxylase is the main products are involved in the mesophyll cells. PEP carboxylase is converted into oxaloacetate. Oxaloacetate means that is the 4-carbon group. Oxaloacetate is converted into malate. Malate it's enter into the bundled sheet cells. CO2 it's enter into the Calvin cycle. Calvin cycle it's sugar, it's came out vascular tissue. At the time this produce a 4-carbon compound, it's involved in the C4 photosynthesis. This 4-carbon molecule then moves into the bundled sheet cell via plasmodesmata. Then in the bundled sheet cell the CO2 is released, the Calvin cycle begins. So, these are the photorespiration, I mean C4 plants, that is the mesophyll cells and the bundle seed cell. So, this is the two cells are involved in the C4 plants. Next photosynthesis. Photosynthesis is nothing but a autotrophic organism use light energy to make the light energy is converted into oxygen gas from carbon dioxide and water. So, this is the definition of photosynthesis. Photosynthesis it's occur in plants, algae and some eukaryotics. Then small molecule combined then stored as a carbohydrate in their bodies. So, photosynthesis that 6 CO2 plus 6 H2O it's converted into light energy, it's converted into C6H12O6 plus 6 O2. So, this is the product of photosynthesis. Next, plant pigment. The principle of plant pigments are chlorophyll, carotenoids, xanthophyll and petalines. anthocyanins. So these are the plant pigments are involved, the main principal plant pigments in photosynthesis. Next, primary functions of plant pigments in plants. So many functions are involved in the plant pigment, like primary function photosynthesis use green pigment chlorophyll along with the red and yellow pigments involved in this session help to capture as much light as possible. Then other function include attracting insects to flowers to encourage pollination. These are the primary function photosynthesis steps are involved. Next, chlorophyll. This is the primary plant pigment in plant. Chlorine absorbs yellow and blue wavelength. Yellow and blue wavelength are involved in chlorophyll, reflecting green. Then all land plants and green algae have two types of chlorophyll. Then chlorophyll A and chlorophyll B. Then heterocotyns contains chlorophyll C. Chlorophyll serves as a fuel of photosynthesis. So these are the types of also involved in the primary pigment in plant. Carotenoids. Carotenoids is nothing but a red, green or yellow pigments are involved in that carotenoids. Functions as accessory pigments in plants. Absorb wavelength not rapidly absorbed by the chlorophyll. For example, found in carrots. Then yellow pigment found in fruits and vegetables. Then lycopene in red pigment in tomato. Role of carotenoids. Attract pollinator and seed disperse. Accessory photosynthetic pigment in periods in low light conditions. Then absorb excessive light energy also induced in carotenoids. These are the main roles are involved in carotenoids. Then antioxidants roles are involved. Then substrate for hormones. So these are the main category also involved in carotenoids, like antioxidants, substrate of hormones. So these are the haploid plant production, micropropagations, then PTC, applications of PTC. So these are the content of this slide. Okay, thank you.